Essentials of Human Anatomy and Physiology. Chapter 3 Cells. Cells and tissues carry out all chemical activities needed to sustain life. Cells are the building blocks of all living things. Tissues are groups of cells that are similar in structure and function. Slide 3.1. Anatomy of the cell. Cells are not all the same. All cells share general structures. Cells are organized into three main regions: nucleus, cytoplasm, plasma membrane. Figure 3.1a. Slide 3.2. The nucleus controls center of the cell. Three regions. Contains genetic material, DNA. Three regions: nuclear membrane, nucleus, chromatin. Figure 3.1b. Slide 3.3. Nuclear membrane. Barrier of nucleus consists of a double phospholipid membrane. Contain nuclear pores that allow for exchange of material with the rest of the cell. Slide 3.4. Nucleoli. Nucleus contains one or more nucleoli. Sites of ribosome production. Ribosomes then migrate to the cytoplasm through nuclear pores. Slide 3.5. Chromatin composed of DNA and protein scattered throughout the nucleus. Chromatin condenses to form chromosomes when the cell divides. Slide 3.6. Plasma membrane. Barrier for cell contents. Double phospholipid layer. Hydrophilic heads. Hydrophobic tails. Other materials in plasma membrane: protein, cholesterol, glycoproteins. Slide 3.7a. Plasma membrane. Figure 3.2. Slide 3.7b. Plasma membrane specializations. Microvilli. Finger-like projections that increase surface area for absorption. Figure 3.3. Slide 3.8a. Plasma membrane specializations. Membrane junctions. Tight junctions. Desmosomes. Gap junctions. Figure 3.3. Slide 3.8b. Cytoplasm. Material outside the nucleus and inside the plasma membrane. Cytosol. Fluid that suspends other elements. Organelles. Metabolic machinery of the cell. Inclusions. Non-functioning units. Slide 3.9. Cytoplasmic organelles. Figure 3.4. Slide 3.10. Cytoplasmic organelles. Ribosomes. Made of protein and RNA. Sites of protein synthesis. Found at two locations. Free in the cytoplasm. Attached to rough endoplasmic reticulum. Slide 3.11. Cytoplasmic organelles. Endoplasmic reticulum, ER. Fluid-filled tubules for carrying substances. Two types of ER. Rough endoplasmic reticulum. Studded with ribosomes. Site where building materials of cellular membrane are formed. Smooth endoplasmic reticulum. Functions in cholesterol synthesis and breakdown, fat metabolism, and detoxification of drugs. Slide 3.12. Cytoplasmic organelles. Golgi apparatus. Modifies and packages proteins. Produces different types of packages. Secretory vesicles. Cell membrane components. Lysosomes. Slide 3.13a. Cytoplasmic organelles. Figure 3.5. Slide 3.13b. Cytoplasmic organelles. Lysosomes. Contain enzymes that digest non-usable materials within the cell. Peroxisomes. Membranous sacs of oxidase enzymes. Detoxify harmful substances. Break down free radicals, highly reactive chemicals. Replicate by pinching in half. Slide 3.14. Cytoplasmic organelles. Mitochondria. Powerhouses of the cell. Change shape continuously. Carry out reactions where oxygen is used to break down food. Provides ATP for cellular energy. Slide 3.15. Cytoplasmic organelles. Cytoskeleton. Network of protein structures that extend throughout the cytoplasm. Provides the cell with an internal framework. Slide 3.16a. Cytoplasmic organelles. Cytoskeleton. 3 different types. Microfilaments. Intermediate filaments. Microtubules. Figure 3.6. Slide 3.16b. Cytoplasmic organelles. Centrioles. Rod-shaped bodies made of microtubules. Direct formation of mitotic spindle during cell division. Slide 3.17. Cellular projections not found in all cells used for movement. Cilia moves materials across the cell surface. Flagellum propels the cell. Slide 3.18. Cell diversity. Figure 3.7, 1, 2. Slide 3.19a. Cell diversity. Figure 3.7, 3. Slide 3.19b. Cell diversity. Figure 3.7, 4, 5. Slide 3.19c. Cell diversity. Figure 3.7, 6, 7. Slide 3.19d. Cellular physiology, membrane transport. Membrane transport, movement of substance into and out of the cell. Transport is by 2 basic methods. Passive transport. No energy is required. Active transport. The cell must provide metabolic energy. Slide 3.20. Solutions and transport. Solution, homogeneous mixture of 2 or more components. Solvent, dissolving medium. Solutes, components in smaller quantities within a solution. Intracellular fluid, nucleoplasm and cytosol. Interstitial fluid, fluid on the exterior of the cell. Slide 3.21. Selective permeability. The plasma membrane allows some materials to pass while excluding others. This permeability includes movement into and out of the cell. Slide 3.22. Passive transport processes. Diffusion. Particles tend to distribute themselves evenly within a solution. Movement is from high concentration to low concentration, or down a concentration gradient. Figure 3.8. Slide 3.23. Passive transport processes. Types of diffusion. Simple diffusion. Unassisted process. Solutes are lipid-soluble materials or small enough to pass through membrane pores. Slide 3.24a. Passive transport processes. Types of diffusion. Osmosis, simple diffusion of water. Highly polar water easily crosses the plasma membrane. Facilitated diffusion. Substances require a protein carrier for passive transport. Slide 3.24b. Diffusion through the plasma membrane. Figure 3.9. Slide 3.25. Passive transport processes. Filtration. Water and solutes are forced through a membrane by fluid, or hydrostatic pressure. A pressure gradient must exist. Solute-containing fluid is pushed from a high-pressure area to a lower-pressure area. Slide 3.26. Active transport processes. Transport substances that are unable to pass by diffusion. They may be too large. They may not be able to dissolve in the fat core of the membrane. They may have to move against a concentration gradient. Two common forms of active transport. Solute pumping. Bulk transport. Slide 3.27. Active transport processes. Solute pumping. Amino acids, some sugars and ions are transported by solute pumps. ATP energizes protein carriers, and in most cases, moves substances against concentration gradients. Slide 3.28a. Active transport processes. Figure 3.10. Slide 3.28b. Active transport processes. Bulk transport. Exocytosis. Moves materials out of the cell. Material is carried in a membranous vesicle. Vesicle migrates to plasma membrane. Vesicle combines with plasma membrane. Material is emptied to the outside. Slide 3.29a. Active transport processes. Figure 3.11. Slide 3.29b. Active transport processes. Bulk transport. Endocytosis. Extracellular substances are engulfed by being enclosed in a membranous vesicle. Types of endocytosis. Phagocytosis, cell eating. Pinocytosis, cell drinking. Slide 3.30a. Active transport processes. Figure 3.12. Slide 3.30b. Cell life cycle. Cells have two major periods. Interphase. Cell grows. Cell carries on metabolic processes. Cell division. Cell replicates itself. Function is to produce more cells for growth and repair processes. Slide 3.31. DNA replication. Genetic material duplicated and readies a cell for division into two cells. Occurs toward the end of interphase. DNA uncoils and each side serves as a template. Figure 3.13. Slide 3.32. Events of cell division. Mitosis. Division of the nucleus. Results in the formation of two daughter nuclei. Cytokinesis. Division of the cytoplasm. Begins when mitosis is near completion. Results in the formation of two daughter cells. Slide 3.33. Stages of mitosis. Interphase. Prophase. No cell division occurs. The cell carries out normal metabolic activity and growth. Prophase. First part of cell division. Centromeres migrate to the poles. Slide 3.34a. Stages of mitosis. Metaphase. Spindle from centromeres are attached to chromosomes that are aligned in the center of the cell. Slide 3.34b. Stages of mitosis. Anaphase. Telophase. Daughter chromosomes are pulled toward the poles. The cell begins to elongate. Telophase. Daughter nuclei begin forming. A cleavage furrow, for cell division, begins to form. Slide 3.35. Stages of mitosis. Figure 3.14, 1. Slide 3.36a. Stages of mitosis. Figure 3.14, 2. Slide 3.36b. Protein synthesis. Gene, DNA segment that carries a blueprint for building one protein. Proteins have many functions. Building materials for cells. Act as enzymes, biological catalysts. RNA is essential for protein synthesis. Slide 3.37. Protein synthesis. Gene, DNA segment that carries a blueprint for building one protein. Proteins have many functions. Building materials for cells. Act as enzymes, Biological catalysts. RNA is essential for protein synthesis. Slide 3.37. Role of RNA transfer. RNA, tRNA, ribosomal RNA, RNA, messenger RNA. Transfers appropriate amino acids to the ribosome for building the protein. Ribosomal RNA, RNA, helps form the ribosomes where proteins are built. Messenger RNA carries the instructions for building a protein from the nucleus to the ribosome. Slide 3.38. Transcription and translation. Transfer of information from DNA's base sequence to the complementary base sequence of mRNA. Translation. Base sequence of nucleic acid is translated to an amino acid sequence. Amino acids are the building blocks of proteins. Slide 3.39.